Hi, I'm Dr. Celeste Allaband, and I'm a fourth-year biomedical sciences student here at UC San Diego. I'm in the Rob Knight and Amir Zarimpar laboratories, and I study the microbiome. We live in an interconnected web of life, not just externally in our environment, but also within our bodies. You are never alone. You will always have the trillions of microscopic organisms of our microbiome that rely on you to survive. You are their entire world. Within our bodies, there are multiple microscopic ecosystems that contribute to our health. These ecosystems are critical to all sorts of physiology, from digestion and metabolism to anxiety and depression. The biggest and most diverse ecosystem of our bodies is in the gastrointestinal tract, which is like the Amazon rainforest of your microbiome. But instead of having a wide variety of plants and animals, it has a lot of different bacteria, fungi, and viruses. You might be asking, how do probiotics contribute to this ecosystem? They are thought to be beneficial, but what they actually do is still unclear. We are just beginning to figure out how this ecosystem works. Probiotics are typically more like tourists. They stick around for a while, they see the sights, but ultimately leave after a few days. They do not typically become permanent members of the microbiome. One of the key questions here is why? Why are some bacteria tourists and why are some permanent residents? This is a really interesting question for me, so in order to research that question further, I set up a rave in the gut Amazon. I took probiotic bacteria and labeled them, kind of like giving them green glow bracelets. The green fluorescence comes from a protein discovered in jellyfish called GFP. I am tracking these bacteria over time while they live inside of a mouse, but this type of relationship between a host and their bacteria is relevant to many organisms. This is important in aquaculture for managing disease. It is important for human and animal health. Let me show you how I would look at these bacteria. I take the poop from the mouse and then add a little water, blending it together. Next, I add a small volume of my sample and plate it onto my petri dish. I add sterile glass beads and then shake the plate to help spread everything around. Now it is ready to grow in the incubator overnight. The next day, I take the plate out of the incubator and look to see if any of my glowing party bacteria are there. If I can't find them on the plates, why did they leave the party early? If they are there, why did they stay? If I can figure out which factors are important, I can build a better probiotic in the future. I could take a city slicker laboratory strain microbe and give it all the tools it needs to become adapted to a new host environment. Then I can start giving them other things to bring to the party, like medicine. Thank you for joining me in the lab. Please look forward to our publication on this work in the future.